Bismillahir Rahmanir Rahim. Welcome again to Derma Immunology. I'm Naglaa Elmongy, Professor of Dermatology, Cairo University. Our video today will be the second video about skin fungi in health and disease and the role of interleukin 17. And again, my main reference will be the same nice review that was published in Parasite Immunology, February 2023, and its title is New Insights into Immunity to the Skin Fungi Shape Our Understanding of Health and Disease. Skin fungal diseases rank among the top 10 most widespread diseases globally, affecting an estimated 20 to 25% of the world's population. The most prevalent fungal skin infections are typically caused by dermatophytes, commonly presenting in humans as tinea capitis, tinea betis, or onychomycosis. And out of approximately 50 known dermatophyte species, trichophyton and epidermophyton species are the ones known to infect humans. And in addition to dermatophytes that primarily infect humans, zoophilic dermatophytes often cause fungal skin infections in animals, particularly in cattle, can be transmitted to humans. Individuals with immature immune system, like in children, and also the elderly with compromised immune defenses, and the immunocompromised individuals such as transplant recipients, cancer patients, and those with HIV infections are typically more susceptible to the development of mucocutaneous mycosis. They are also vulnerable to a wider spectrum of fungal pathogens, and this highlights the key role of the immune system in preventing excessive fungal growth and tissue invasion. Therefore, mucocutaneous candidiasis frequently occurs in individuals receiving immunosuppressive treatments related to cancer or transplantation, as well as those with acquired or hereditary immune deficiencies, such as AIDS patients or individuals with genetic mutations in specific T helper cell related genes. This highlights the specific importance of T cell mediated immunity in controlling fungal infections in barrier tissues. The significance of an intact skin barrier in protecting against fungal infections is highlighted by the fact that various fungal pathogens take advantage of injured skin sites to breach this protective barrier. Consequently, skin infections caused by species like Candida or Aspergillus or others are common and of significant concern in individuals who have suffered burns. These fungi, among others, can also lead to complications in chronic wounds, exacerbating the healing process, as seen in conditions like diabetic foot ulcers. Chromoblastomycosis, eumycetoma, and sporotrichosis may develop when fungi enter the skin through accidental means such as trauma or surgical implantation, resulting in subcutaneous lesions if not timely treated. The common skin commensal yeast, malassezia, has also been linked to various skin disorders, including dandruff, pityriasis versicolor, seborrheic dermatitis, and atopic dermatitis. Evidence of a causal connection between malassezia and certain conditions has emerged through observations that antifungal treatments can relieve symptoms, leading to improvement in dandruff and seborrheic dermatitis. However, the role of malassezia in the pathogenesis of atopic dermatitis remains somewhat uncertain. Fungal skin infections are typically mild and can be effectively treated, although achieving complete resolution may require a long and sometimes challenging process. In immunocompromised individuals, managing fungal infections is often more complex and the disease course tends to be chronic or marked by recurring episodes. One of the challenges in achieving effective antifungal treatment is the limited availability of pharmacological drugs, in addition to the emergence of resistance to existing compounds. Now I'm going to talk about defense mechanisms of the skin against fungal infections. Although fungal infections are becoming a growing medical concern, they are relatively uncommon despite the constant presence of fungi in the environment and on the skin microbiota. This leads to the question of how the skin manages to control fungal presence during normal balanced conditions, as well as the conditions under which fungal skin diseases can still develop. The first host defense against fungal infection is the skin barrier. The skin is a protective shield that defends the body against external threats by establishing a physical barrier and serving as the framework for a complex immune system. The ongoing renewal of the epithelium and the healing of wounds following injury are fundamental processes that keep the integrity of this shield. And in addition to providing physical protection against fungi, epithelial cells have the ability to detect and distinguish various levels of fungal virulence. When faced with these variations, they trigger an immune response cascade. Epithelial cells play a key role in producing antimicrobial peptides, which can inhibit or be toxic to fungi. As regards the role of immune system in host defense against fungi of the skin, immune cells residing in the skin's epidermis and dermis are strategically located to effectively monitor and ensure efficient immune surveillance of skin fungi, preventing excessive growth and tissue invasion. They do this by directly detecting fungi and their products through pattern recognition receptors and responding to alarm signals released by keratinocytes. Langerhans cells, which create a network within the epidermis, have been demonstrated to aid in the monitoring of Candida albicans by serving as antigen-presenting cells. In the dermis, several types of tissue-resident immune cells are found, including macrophages, mast cells, various subtypes of dermal dendritic cells, as well as gamma delta and alpha beta T cells. These cells play a vital role in maintaining immune balance when living with commensal fungi. They also have a key function in generating protective immunity against fungi that invade the skin. This is achieved through the production of cytokines and antimicrobial responses. Also, when the skin is invaded by fungi, circulating immune cells are quickly recruited to the site. The interaction between skin fungi in cutaneous well-being and disease is complex. Immunity to skin fungi varies depending on the situation, with fungal elements capable of modifying the host's immune response, impacting health when in a commensal state, and contributing to disease during infection and immunopathological conditions. Dendritic cells primarily detect commensal fungi through C-type lectin receptors, or TRPV1 positive neurons. This detection leads to the recruitment of interleukin-17A producing gamma delta T cells and the initiation also of adaptive immunity to specific fungi. This includes the presence of detectable immunoglobulin G in the blood, as well as the development of local and systemic T helper 17 cells. Gamma delta T17 and T helper 17 play a role in maintaining fungal balance during commensal state through the secretion of interleukin 17A and interleukin 22. Also, the cutaneous microbiota interacts with the skin fungi in several ways, including gene acquisition via horizontal gene transfer or malassezia-mediated inhibition of Staphylococcus aureus biofilm formation. When the epithelial barrier is broken or when fungi enter through wounds, they can infiltrate deeper tissue layers, leading to inflammation mediated by interleukin-17 and causing additional harm. The secreted aspartyl protease 1 of Malassezia flavor has the potential to disturb the wound healing process. In case of innate immune deficiencies affecting interleukin-17 induction, including those related to CARD9, the downstream adapter in C-type Lecter receptor signaling, the skin ability to manage fungal presence becomes compromised, and this can lead to fungal overgrowth and the development of chronic mucocutaneous candidiasis as a consequence. On the other side, an excessive or pathogenic production of interleukin-17 can result in chronic inflammation and skin changes as seen in psoriasis. Also, apparent antifungal type 2 responses, including fungus-specific T helper 2 and IgE, are recognized in atopic dermatitis. In cases of atopic dermatitis flares, there is typically a compromised skin barrier and an increase in Staphylococcus aureus. Also, the severity of atopic dermatitis is linked to specific type 2 immune responses directed against Malassezia. Allergens originating from Malassezia, such as Mala S11 and Mala S13, can serve as autoantigens because of their ability to cross-react with host proteins, thereby exacerbating the disease. Metabolites and enzymes produced by fungi have the potential to cause skin irritation or disrupt the skin barrier function in the host. Also, the conserved components of the fungal cell wall, like beta-glucans, are critical to immune recognition and the initiation of protective antifungal responses. Lastly, I hope that you have enjoyed my second video about skin fungi in health and disease, and I'm reminding you again to subscribe to the channel and activating the alarming bell, and thank you.